almost as soon as the pandemic was declared, the buzz around on social media was, oh, there was going to be a vaccine that's going to edit our genes. I didn't think much of it, not until I found the patent about human-robot new species in July, right at the beginning of July, that I started to think, well, you know, maybe there's something to this. But then with everything else that's happened, I didn't have time to look into it anymore. Recently I've been looking at the ingredients that were declared on Monday for the COVID vaccine and comparing those to the declared ingredients on the patent, and I didn't think it would take me very long, a couple of days to research, prepare, do the voice and get it up. But there are so many questions, so many discrepancies, it's taken me much longer. However, off the back of that, I found the lipid nanoparticle articles that I was reading were, not all of them, but I'd say half of them were talking about genome editing. So I started to think about that maybe there was something to it. What I found really surprised me, because I didn't think it would be possible to edit genes intramuscularly, but apparently it is, and I'll show you what I found and see what you think. To make it really simple, genome editing is a two-step process which starts by filling cells with DNA that encodes for genome editing proteins, which then begin to alter the genome. I'm only going to show you two examples. This was never going to be a long video with lots of detail. This was merely to draw your attention to the fact that, yes, intramuscular genome editing can be done. The first example is an in vivo experiment regarding a mouse. I'll be referring to the substances used as substance 1 and substance 2, just to keep it really simple. Now, substance 1 contained cationic lipids, DOTAP. You'll hear me talk about these in my next video about the vaccine. DOTAP lipids are used in that. It's the same delivery system in both the gene editing and the COVID vaccine. That was injected into the mouse's leg. That was substance 1. Substance 2 was also injected to the mouse's leg, but this substance didn't have the cationic lipids, the DOTAP. Whichever substance showed up tissues with red fluorescence was a substance that was editing the gene, and the results were that substance 1 had the higher fluorescence. It was red fluorescence, and it proved that gene editing was in progress. The purpose of the CRISPR strategies are to alter the genome permanently, but the problem is the durability and the side effects are still unknown, and this article that I'm using was written in February 2019. The second and final example of intramuscular genome editing that I'm going to show you examines just that, the durability and the side effects. The method used this time wasn't the lipid nanoparticle RNA method, it was viral vectors plus CRISPR. Now, viral vectors in this case were the adeno-associated viruses, and if you think of them as a mixer, so you would mix the AAV with a virus that you want to create the immune response to. So AAV plus measles, AAV plus HIV, or even plus COVID-19, that kind of thing. It's just a mixer. So the researchers looked at restoring the expression of the protein dystrophin in a mouse model of DMD, which is Duchenne muscular dystrophy. And if you don't know what that is, it's only affects boys. It's a muscle wasting disease, really cruel, and it affects the X chromosomes. The results were as follows. Total genome editing in adults was not sustained between 8 weeks and 6 months post-injection. But in newborns, it showed sustained editing in the heart, the diaphragm, and the tibialis anterior, and that is the muscle in the outside of the lower leg. The most important side effect that came out of this experiment, this research, was the fact that genomes were edited that weren't supposed to be edited, which is obviously a major issue, and it needs to be addressed in further studies. So these are just two examples. I wanted to show you that intramuscular genome editing is possible. However, It's only been done once in humans in a trial, and it didn't have a good outcome. One of the recipients died. So do I think that this is what's happening with the COVID-19 vaccine? The short answer is no, I don't. And not only because of the research that has only been done successfully-ish in mice, it's also because of the age range of the people who first had the vaccine. It was the over 80s. Now, what would be the point of editing genes in someone who's over 80 and, without sounding too blunt, coming towards the end of their lives? What would be the point? If they'd started with the younger people, like the mid-teens to the mid-30s, I would have said, yeah, it's possible, because there's all these ideas that they want to depopulate the world, and that would make more sense if they'd picked on the younger people. But to start with the older people, no, I don't think it is to do with the genome editing. I do, however, think it's to do with control. I think the whole point of the virus was for us to have the vaccine, to do with monitoring us, collecting our data, and to merge us with AI. Now, when I say that, I don't mean turn us into a robot. I mean to make us reliant on technology. Just that. I mean super reliant, not just in recreational ways, but to live our daily lives. So that's what I think. As I said at the beginning, I am going to put another video up. If not today, it'll be tomorrow. Done all the prep, just got to do the voice. It's about the vaccine. The declared ingredients don't. There's some. I knew there was something wrong when I saw the list. It says including the lipids, including such and such and such. And when they say including, it means they're not telling us everything. So that's why I went to the patent and thought I'd get it, because they have to declare what's in it. As I was reading the patent, I realised that there were a lot of vague parts. It said things like lipid-like, phosphoid-like, without actually saying what this substance was. And it wasn't just me. At the end, there's some legal points on the patent, and the patent office has said, well, this part is unclear, this part doesn't say really what's going on. So that's what my next video is going to be. It's going to be working that out, and why haven't they told us that this is in it. One of the declared acids was valproic acid. Now, you probably heard of it. It was associated with autism and thalidomide. At the moment, I haven't actually found it in any form, so I don't understand why it's on their list, but I'm hopefully going to clear that up by the time I put the video out. If not, I will just mention it and say I couldn't find any evidence of it, but it is declared. Thanks for watching.